Meiosis is a type of cell division that occurs in the reproductive cells of an organism to produce haploid gametes like egg and sperm. Meiosis is similar in some ways to mitosis, which you should already be familiar with, but there are also some key differences which you will see. Just like mitosis, meiosis progresses through phases of division: prophase, metaphase, anaphase, telophase, and cytokinesis. The difference is that mitosis only involves one division, while meiosis involves two divisions. So meiosis will progress through the phases of division twice, while mitosis only does once. Meiosis also produces 4 haploid cells, while mitosis results in only 2 diploid cells. Let's take an in-depth look at the events of meiosis so you can see the similarities and differences for yourself. The first division of meiosis, meiosis 1, begins with a diploid cell in interphase. Don't forget that during interphase, the chromosomes are replicated so that there are 2 copies of each. During prophase 1, the chromatin has condensed into chromosomes. The nuclear envelope breaks down, and the spindle fibers start to develop as the centrioles migrate to opposite ends of the cell. Chromosomes that are matching in size, shape, and genetic content are called homologous chromosomes. During prophase 1, homologous chromosomes pair up and spend the rest of meiosis 1 moving together like dance partners. Spindle fibers attach to the centromeres of the homologous pairs. During metaphase 1, the homologous pairs line up along the equator of the cell. Their movement is directed by the centrioles and spindle fibers. During anaphase 1, the spindle fibers pull the homologous chromosomes apart so they are separated from their dance partner. Telophase 1 and cytokinesis occur as the chromosomes reach the poles of the cell and the cytoplasm divides. Meiosis 1, the first division, is now complete, and we are left with 2 haploid cells that will enter the second division, meiosis 2. Meiosis 2 begins with prophase 2. The centrioles divide and move to opposite ends of the cell. The spindle fibers appear and attach to the centromeres of the chromosomes. During metaphase 2, the chromosomes line up along the equator of the cell, directed by the spindle fibers and centrioles. Anaphase 2 occurs as the spindle fibers pull the sister chromatids apart and the 2 identical copies of the chromosome move away from each other towards opposite ends of the cell. During telophase 2 and cytokinesis, a nuclear envelope forms around each group of chromosomes. The spindle fibers disappear, and the chromosomes begin to unravel back into chromatin. The cytoplasm divides, and we are left with 4 haploid cells that are genetically different from each other and from the parent cell. Meiosis results in a huge amount of genetic variation amongst the cells it produces. First of all, the way the homologous chromosome pairs line up during metaphase 1 is completely random, so each time an organism performs meiosis, the chromosomes could line up differently. In this example, we're looking at a hypothetical organism with 4 chromosomes in each of its cells. After replication, each cell would have 2 copies of each chromosome. During prophase 1, the homologous chromosomes pair up, and during metaphase 1, the homologous pairs line up along the equator of the cell. Because the movement of the chromosomes is random, the homologous pairs could end up arranged like this or like this. Either arrangement is equally possible. When homologous pairs separate or segregate during anaphase 1, the chromosomes are sorted out into 2 groups that enter the 2 newly forming cells. Since the chromosomes line up randomly during metaphase 1, the chromosome groupings made during anaphase are different each time, and the resulting gametes are different as well. This concept is known as independent assortment, which you might recall is one of Mendel's laws. The movements of the chromosomes during meiosis explain why many traits are inherited independently of each other, as Mendel's law states. Independent assortment creates a huge amount of genetic variety all by itself, but there are additional forces at work that provide even more opportunity for genetic variation. Crossing over is a process during which homologous chromosomes can exchange genetic information while they are paired up during prophase 1 of meiosis. Essentially, two homologous chromosomes cross segments, and then the segments trade places so each chromosome leaves with sections of the other. It provides a whole new realm of genetic possibilities for gametes. The number of different genetic combinations an organism can make by meiosis varies depending on how many chromosomes the organism has. A human, for example, has 23 pairs of chromosomes, or 46 chromosomes total. With that number of chromosomes, a human individual can make a total of 8,388,608 different gametes by independent assortment alone. Once you factor in the process of crossing over, the possibilities truly become limitless. While the genetic variation created in gametes is huge, when you consider the randomness of fertilization and which sperm will fertilize which egg, the variety of genetic combinations becomes mind-boggling. Meiosis can be used to create haploid spores in plants and haploid gametes in animals. Let's take a closer look at gametogenesis in animals, the process of making egg and sperm through meiosis. In male animals, the process of making sperm, or spermatogenesis, occurs in the testes. The process begins with diploid cells called spermatogonia, which divide by mitosis to produce primary spermatocytes. The primary spermatocytes undergo the first meiotic division to produce two haploid secondary spermatocytes. The secondary spermatocytes continue through the second division of meiosis to produce spermatids that will develop into mature sperm. Once a male individual reaches sexual maturity, he can produce an unlimited number of sperm by this process. In females, the process of making eggs, or oogenesis, occurs in the ovaries. Unlike males, females are born with a finite number of reproductive cells called oogonia. While males can produce unlimited amounts of sperm, females have all the oogonia they will need at birth and cannot produce any more during their lifetime. Shortly after birth, the diploid oogonia develop into primary oocytes, which are also diploid. Their development stops at this stage until the female reaches sexual maturity. After puberty, the primary oocyte resumes meiosis. The primary oocyte divides unequally, producing a large secondary oocyte and a much smaller polar body. Both the secondary oocyte and the polar body are haploid cells. Each cell goes through the second meiotic division. The secondary oocyte undergoes another unequal division to produce a large ootid and another polar body. The ootid will eventually develop into a mature ovum or egg cell that will be released from the ovary during ovulation. The three polar bodies break down and deteriorate. Sexual reproduction could not occur without the process of meiosis. The beauty of this type of cell division is that it creates so much genetic variation within a population. Genetic variation is pivotal to the survival of a species, so sexual reproduction gives organisms a competitive edge in the process of evolution.